Welcome to this next video in which we are discussing tuberculosis and anti-tuberculosis drugs. Okay, so in the previous video we discussed the Mycobacterium tuberculosis, and we spent a long time looking at the structure of the Mycobacterium tuberculosis bacterial cell wall. In this video what we're going to do now is actually begin the story of tuberculosis, begin the story of being infected by this bacterium and what's actually going to happen. So we're going to begin with transmission. So we'll go over onto the other side and we'll begin with transmission. So how do you actually catch tuberculosis? Well, you catch it from other people who currently have an active tuberculosis infection. So remember, although I haven't explained it properly, I've told you about how there are these two different types of TB infection. You can either have an active TB infection or a latent TB infection. Now, if you have a latent TB infection, then it's not actually causing any disease in you at the moment, and also you are not capable of spreading it to other people. You have to have an active tuberculosis infection in order for you to be able to spread it to other people. So let's just draw some little stick men here. So let's have person 1 here. Okay, so here's stick man number 1, and let's say that stick man number 1 is actually infected with tuberculosis, and let's say that poor old stickman number 1 has actually got an active tuberculosis infection, so he is not very well at all. Okay, now when you have an active TB infection, you're going to be coughing a lot. That's one of the major symptoms of an active TB infection in the lung, that it's going to be irritating the lung and that will trigger you to cough. And you'll cough up a lot of sputum which will contain Mycobacterium tuberculosis bacterial cells. So when person 1 coughs, he's going to be propelling into the air lots of Mycobacterium tuberculosis bacterial cells. So let's say that person 1 now does cough, which I'll show by this arrow here, and what's going to happen is tiny little Mycobacterium tuberculosis cells are now going to be in little droplets in the air. So I'm going to colour these in red, because remember, if you stain them with the Zeal-Nielsen stain, they appear red, so it's kind of like the classical colour to show a Mycobacterium tuberculosis cell. Okay, so person 1 who has an active TB infection, and don't worry, we will see, by the end of this video, I promise you will understand what the difference between an active TB infection and a latent TB infection is. But person 1 at the moment has this active TB infection, and he has coughed and has propelled lots of Mycobacterium tuberculosis cells into the air. Now, let's have stickman number 2 here. So over here is stickman number 2, and let's say that stickman number 2 has never ever been exposed to Mycobacterium tuberculosis before. So they don't have an active TB infection, and they don't even have a latent TB infection. They've never been exposed to tuberculosis before. So we're going to look at the story for stickman number 2 here from the beginning. And when you get infected with tuberculosis for the first time, this is what's known as a primary TB infection. Okay, so how is stickman number 2 then going to actually become infected? Well, of course, he's going to breathe in some of this air that has Mycobacterium tuberculosis bacterial cells within it, and therefore these cells will be brought into his lungs. And this is the reason that tuberculosis usually begins as a lung infection, because this is the primary site where the bacterial cells first arrive. If you know anything about the atypical causes of tuberculosis, so some of the other types of Mycobacterium which can cause tuberculosis, such as Mycobacterium bovis, you will know that those forms of tuberculosis can begin at other sites in the body, for instance the intestine, because Mycobacterium bovis is generally caught from milk of cows that have got Mycobacterium bovis, and therefore the first site that it arrives is the intestine rather than the lungs. But as I say, that's a very atypical form of tuberculosis, and we're not going to dwell on that in this video. So our form of tuberculosis, the major form of tuberculosis, is going to begin in the lungs, because that's where the bacterial cells first arrive. So, what's going to happen then? Let's just draw a few little simple pictures. The Mycobacterium tuberculosis is going to come down person 2's trachea here. So, here we are. Here's the trachea, so they'll come down the trachea, and then of course they'll get to the carina, where the trachea bifurcates into the two major bronchi, the right bronchus and the left bronchus here. So I'll just label a little bit of this picture up here. So this is the trachea, and then the bifurcation of the trachea is known as the carina, and then we've got the right bronchus and the left bronchus, and these cells will go either of these two ways. And of course they'll go through smaller and smaller bronchi, then they'll go into the bronchioles, and then they'll finally get down into the alveolar ducts and then into alveoli. And that's the picture that I want to now show you. So I'm going to show you an alveolar duct firstly. So the tiny little vessels of the lung are known as alveolar ducts, and these are the little airways where the actual walls of these airways are made up by alveoli. So I'm drawing an alveolar duct here. So these are the outpouchings, these are the alveoli, and you can see that the tube down the middle here, which is known as the alveolar duct, it's really just made up by alveoli. The walls of this alveolar duct are just made up by alveoli. So this little outpouching here, this is an alveolus, and the alveolar duct is just made up by loads of alveoli sort of forming a tube. So eventually, if you follow these tubes for long enough, if you were a little man, or indeed if you were a little Mycobacterium tuberculosis cell, if you were to follow your way through the tracheobronchial system for long enough, if you were to go down the tubes for long enough, you would eventually get to alveolar ducts and then into alveoli. And what I now want to do is draw a more in detail picture of some of these alveoli here, and actually see what's going to happen to the Mycobacterium tuberculosis when it arrives in these alveoli. So let me just draw a little part of the alveolar duct in more detail. So here's a little part of the wall of the alveolar duct. So here are two alveoli, one alveolus, a second alveolus, and now I'll just have them lined by pneumocytes. And remember, there are two different types of pneumocytes. There are squamous pneumocytes known as type 1 pneumocytes, and then there are much taller pneumocytes known as type 2 pneumocytes, of which I've just drawn one there, which are the pneumocytes that actually produce the surfactant, which is really important for keeping the airways from collapsing, well, keeping the alveoli from collapsing because of the surface tension of the fluid on the surface of the pneumocytes. Okay, so I'll just colour a little bit of this in. So all of these pneumocytes will of course be on a basement membrane, so I'll just show this basement membrane in pink here. This is what keeps them in position. And as I say, we've drawn two different types of pneumocytes here. The squamous ones are known as type 1 pneumocytes, so this is a type 1 pneumocyte, and obviously pneumo refers to the respiratory system, so this is a cell of the respiratory system, and these make up most of the surface area of the alveoli because of how flat they are. So they have much greater surface area than, for instance, the type 2 pneumocytes. This one here, this is representing a type 2 pneumocyte, and type 2 pneumocytes, believe it or not, are actually present in about equal numbers to type 1 pneumocytes, but they make up a much smaller fraction of the surface area of the alveoli simply because they have such a smaller surface area than the type 1 pneumocytes. So this is a type 2 pneumocyte, and its major role, of course, is in secreting the surfactant. Okay, so here is a basic picture of two alveoli here. Now, just before we actually go back to the story of the Mycobacterium tuberculosis arriving here then, let's add on one more important cell. And the other important cell that I want to mention is the alveolar macrophages. So inside alveoli, you have macrophages that are resident within there. So I'll draw one of these here. Now remember, macrophages, they are great blobs of cells. So here, this is representing an alveolar macrophage, And what is the job of these alveolar macrophages? Well, they move from alveolus to alveolus, and they look for any sort of particulate matter that should not be present within those alveoli, and if they find it, they will phagocytose it. So this is an alveolar macrophage, and I'll just colour it in orange, like so. So, ooh, that's smudged horribly, but never mind. So, these alveolar macrophages, they move from alveolus to alveolus to alveolus, so they're not static, they're motile, and they're just looking for particulate matter, or indeed bacterial cells or other pathogens, viruses, fungal cells potentially, maybe even protozoal cells, or potentially even multicellular parasites. They're looking for any sort of pathogen that should not be there, or particulate matter that should not be there, and if it is there, they're going to phagocytose it, break it down and clear it. So they are involved in keeping the alveoli clean and free of dirt and infection. Okay, so those are important cells for us to be aware of, because they are going to be the first cells to realise that there is something very, very bad in the alveoli when we breathe in these Mycobacterium tuberculosis cells. Okay, so back to the story then. So once again, here are the Mycobacterium tuberculosis cells, and we have breathed them in, and now they are coming into our alveoli. So let's actually just draw one down in this alveolus here. So this tiny little rod that I've drawn here and I'm now colouring in red, that is going to represent a Mycobacterium tuberculosis, MTB for short, now in this alveolus. So what we want to ask now is, what's going to happen? Well, the alveolar macrophages are not going to like the fact that this Mycobacterium tuberculosis is now present within this alveolus. But the first question that we need to ask is, how on earth is it going to realise what this thing is? How is it going to recognise that this is something very dangerous? So that's the first thing that I want to explain. How is it going to recognise the Mycobacterium tuberculosis cell as something that is foreign and dangerous? Then what I want to talk about is, what's its response going to be to the fact that it has found something dangerous within the alveoli? Well, I've already told you about that. It's going to phagocytose it, but it's also going to call for help. It's going to initiate the inflammatory response. And I want to now discuss all of this. So, the beginning topic then is recognition. How is the alveolar macrophage going to recognise that this new cell that has come into this alveolus here is a dangerous cell? Okay, well, this is the topic of pattern recognition receptors. So, alveolar macrophages, and indeed loads of other cell types all around the body, have the ability to recognise foreign, dangerous cells. They have the ability to recognise pathogens. And the way they do this is through a whole variety of receptors that overall are clustered under the umbrella term pattern recognition receptors. So if I just draw an alveolar macrophage here, so here is an alveolar macrophage, and I'll just colour it in orange. These cells are covered in a whole bunch of different receptors that we're going to collectively refer to as pattern recognition receptors. And what do these receptors do? Well, they recognise molecules, molecular patterns that no human cell would ever in its right mind make. You see, that's the way that we can actually recognise pathogens, by looking for molecular patterns, molecules that no human cell would ever make. Because if we suddenly see one of these molecules that we know no human cell would ever make, then it's a giveaway sign that whatever it is that has that molecule is not good, it's not human, certainly. So that's the way in which cells of the body can recognise pathogens, indeed the way that these alveolar macrophages are going to recognise the presence of a pathogen in the alveoli. So here, this blob on the surface of the macrophage, this is going to be an example of a pattern recognition receptor, which for short is abbreviated to PRR. So this is a pattern recognition receptor. And this will have as its ligand a specific PAMP, as it's known, which stands for pathogen-associated molecular pattern. Okay, so if I just draw a little pathogen here, so let's say this is our Mycobacterium tuberculosis, on its surface it might have a molecule that no human cell would ever make. So here's the Mycobacterium tuberculosis, and I'll just colour this molecule in blue here. So here, this is the ligand for the pattern recognition receptor, which I'll colour in green, and this collectively is known as a pathogen-associated molecular pattern, and for short that's abbreviated down to PAMP. So in blue here, that blue box is representing the PAMP, standing for pathogen, and then the A is for associated, and then the MP is for molecular pattern. So again, pathogen-associated molecular pattern, it's an umbrella term for a huge number of different molecules. There are loads of molecules that different pathogens that might attack the human body have, which no human cell would ever make. We cluster them all under this great big heading of pathogen-associated molecular patterns. Okay, and we call the receptors for these different pathogen-associated molecular patterns pattern recognition receptors. But I want to stress that it is not just the case that you have, for instance, one pattern recognition receptor which can recognize all PAMPs. That's not the way it works. Instead, this is an umbrella term. You have loads of different pattern recognition receptors, and they all recognize different examples of PAMPs. So to give you an example that's very relevant to Mycobacterium tuberculosis, a pattern recognition receptor that is on the surface of our alveolar macrophages is a receptor known as toll-like receptor 2, which for short is abbreviated down to TLR2. So this stands for toll-like receptor 2. Okay, so TLR2. And the pathogen-associated molecular pattern that toll-like receptor 2 can recognize is actually a molecule that we have discussed already. A molecule that, for this very reason, I brought up. That molecule was lipomannan molecules. So toll-like receptor 2 can actually be activated by lipomannan molecules. So lipomannan molecules on the surface of the Mycobacterium tuberculosis, these combined to toll-like receptor 2 molecules on the surface of the alveolar macrophage, and that is a giveaway sign that will activate an intracellular signaling cascade inside the alveolar macrophage that tells the alveolar macrophage there is something here that is dangerous, okay? And we need to do something about it. So that's the way that the alveolar macrophages can recognize the presence of the Mycobacterium tuberculosis and know that this is not a human cell and that this is dangerous through these pattern recognition receptors which have as their ligand PAMPs. Okay, what I now want to discuss is what's the alveolar macrophage going to do. So once it's found the presence of these Mycobacterium tuberculosis cells, what's it going to do? Well, it has two major responses that I'm going to discuss with you. Firstly, it can phagocytose the Mycobacterium tuberculosis. So we're now on responses. So I'll just put this as a little title here. So firstly, it is capable of phagocytosing the Mycobacterium tuberculosis. And secondly, it's also going to release pro-inflammatory mediators that are going to call for backup. They're going to initiate the inflammatory response. So pro-inflammatory mediators. Okay, so I want to discuss both of these. So we'll start off with phagocytosis. So phagocytosis involves an alveolar macrophage engulfing the Mycobacterium tuberculosis cell into a vacuole inside its cytoplasm known as a phagosome, and then trying to break the Mycobacterium tuberculosis within the phagosome down, so digest it. So let me show you this process. So we'll start off with the engulfing process. So if I show my alveolar macrophage here, let's now have it engulfing the Mycobacterium tuberculosis. So it has recognized the Mycobacterium tuberculosis cell as something that is very bad and potentially very dangerous, and certainly not human. So now what it can do is it can engulf that cell into a vacuole, like so. So here's the Mycobacterium tuberculosis, and you can see that the membrane's invaginating around it. So it's gradually being taken up into a vacuole here. So here's the nucleus of the macrophage again, and I'll just colour the outline of the macrophage in orange again, and also colour the Mycobacterium tuberculosis in red there. Okay, now of course what this will progress to is that invagination, that forming vacuole that we've got there, will be pinched off, and then you'll actually have the Mycobacterium tuberculosis enclosed in a vacuole completely. So this will progress to this picture next. So here is our cell, here's the nucleus, and now inside a vacuole you've got the Mycobacterium tuberculosis. And that vacuole that contains the Mycobacterium tuberculosis cell that has been engulfed in this way is now referred to as a phagosome. So I'll just label it up as a phagosome. So this is the first part in phagocytosis, the actual engulfment of the bacterial cell into a phagosome in the way that I've just described. What now needs to happen is that the Mycobacterium tuberculosis needs to be digested in that phagosome. And I'm afraid this is the point where the story goes a little bit wrong. So what I'm going to tell you is what should happen now, and then what we're going to discuss is what actually does happen. So the reason that Mycobacterium tuberculosis is such a chronic disease, and why once you've been infected with Mycobacterium tuberculosis you never really actually get rid of all the cells in your body, you're always infected with it. Even if you've got a latent infection, it's still there, it's not causing disease. The reason that occurs is that macrophages really struggle to actually digest the Mycobacterium tuberculosis. Mycobacterium tuberculosis cells resist being digested once they're in a phagosome extremely well, and this is why these bacterial cells are not just going to be annihilated when they're breathed into the lung. Okay, so I'm going to outline what should happen, and this will occur in a few macrophages. A few macrophages will get lucky and they will manage to digest their Mycobacterium tuberculosis cells. Others will have less luck, and the Mycobacterium tuberculosis cell will get the better of them. And remember, we haven't just got one Mycobacterium tuberculosis cell coming into our lungs, we've got them all over the place. We breathed in a lot of Mycobacterium tuberculosis cells potentially. So you've got this happening potentially in multiple different sites around the lung. Okay, so let me now outline what should happen. So there are two major processes that should help to destroy the cell inside the phagosome, and they are that we're going to fuse lysosomes with the phagosome to form a phagolysosome, and we're also going to start producing reactive oxygen species and reactive nitrogen species and concentrating them in the phagosome. So let me show these two processes. So firstly, let me talk about lysosomal fusion. So lysosomes then. Lysosomes, as I'm sure you're aware, are vesicles inside the cytoplasm of a cell which contain a huge number of lytic enzymes. So if I just draw a few of these. So let's imagine that we're zooming in on this picture here. So zoom in on the phagosome. So I'll just draw the phagosome much bigger than previously. So this is the phagosome with the Mycobacterium tuberculosis cell inside. Okay. I'll colour it in red like so, and then we're going to have lysosomes also in the cytoplasm. So here, these membrane-bound vesicles here, these are going to represent lysosomes, and lysosomes are absolutely full of enzymes which we can collectively know as lytic enzymes. And these enzymes are going to break stuff down. That's what lytic means. It means to break stuff down. And you'll have lytic enzymes that are capable of breaking down all sorts of different biomolecules. So by biomolecules, I mean nucleic acids, proteins, lipids, carbohydrates. You'll have enzymes for all those different types of biomolecules that will be able to break them down. So break down, for instance, RNA, break down proteins, break down polysaccharides, etc. Okay, so these are incredibly powerful tools that the cell has, and indeed the cell often uses lysosomes as somewhere that it can send molecules that it's finished with to be broken down back into their building blocks, which can then be recycled by the cell and used again. Okay, but here in phagocytosis, what we're going to do is we're going to fuse lysosomes with the phagosome membrane and just release all of these lytic enzymes onto the cell or the pathogen that is present inside the phagosome, and that's one of the key ways that we can then break down that pathogen, because of course these lytic enzymes will just break down their biomolecules and then hopefully the pathogen will disintegrate. Okay, so what's going to happen then is we're going to fuse these lysosomes with the phagosome, and that's known as phagolysosomal fusion. So phagolysosomal fusion will occur, and then the structure that we're left over with afterwards, which will now be a bigger membrane-bound structure, is then called the phagolysosome. So the phagosome will undergo phagolysosomal fusion, where lysosomes will fuse with it, and after that process has occurred, it's then called a phagolysosome. And I'll stress this is what happens in the ideal situation. I'm going to talk in a moment about how Mycobacterium tuberculosis is going to resist this mechanism, but let's say it will occur in a few macrophages. A few macrophages will get lucky and this process will be able to actually proceed. So we've covered phagolysosomal fusion. What I now want to talk about is the other major killing mechanism that we have as part of phagocytosis, which is the production of reactive oxygen species and reactive nitrogen species. So let me begin with the production of reactive oxygen species, and then I'll go on to the production of reactive nitrogen species. So, in the membrane of the phagosome, what you are going to set up is an enzyme. You're going to build an enzyme complex which is capable of producing superoxide, which is the starting point for reactive oxygen species. In addition, you're going to send induced nitric oxide synthase enzymes to the outer portion of the phagolysosome, and they will produce nitric oxide, which is the starting point for reactive nitrogen species. So let me just go back a little bit. The idea with reactive oxygen species and reactive nitrogen species is that we can effectively poison the bacterial cell or the other pathogen that is present inside the phagolysosome. These are incredibly reactive molecules, and I've abbreviated them down here. Reactive oxygen species is often abbreviated down to ROS. So this stands for reactive oxygen species. And then RNS, that stands for reactive nitrogen species. So just take the oxygen out and substitute it in with nitrogen, and then you've got what RNS stands for. Okay, so the principle here is that these are incredibly reactive small molecules based either on oxygen or nitrogen that will react with biomolecules and will cause great damage to cells. They're poisonous to cells, incredibly cytotoxic, these molecules. Too powerful, too reactive, and will just lead to cell death. So the idea is that when we've got some bad cell inside our phagolysosome, we might want to produce these molecules in the phagolysosome so that they can attack the pathogen. And that's indeed what happens in phagocytosis. So what I want to now talk about is how you can actually get the production of reactive oxygen species and how you can get the production of reactive nitrogen species. So let me do it on my picture over here. So what happens in the membrane of the phagolysosome, you assemble an enzyme complex that is capable of producing the starting point for producing all of the reactive oxygen species, which is superoxide, which is itself an extremely powerful reactive oxygen species. So in the membrane of the phagolysosome, you produce an enzyme complex that is known as NADPH oxidase. Okay, and NADPH oxidase is also called phagocyte oxidase. So either of those names can be used for it. Now, this is a multi-protein complex. It's made up of lots of different proteins, and some of the proteins were already in the phagosome membrane, and some of them were in the cytoplasm. The ones that were already in the phagosome membrane, they are just present in the plasma membrane. Now, because they haven't got the other proteins that are in the cytoplasm bound to them, they're not actually active. So you have some of the components of the NADPH oxidase already in the plasma membrane, but they're not doing anything because they haven't formed the full NADPH oxidase complex. When you phagocytose a pathogen in the phagosomal membrane, you will end up with these proteins also being present. What you can now do as part of the killing mechanism for phagocytosis is use these proteins along with other proteins from the cytoplasm to build a full NADPH oxidase complex, which is also known as the phagocyte oxidase enzyme complex. So the important overall message here is that in the membrane of the phagosome or the phagolysosome, depending on which point of the lysosomal fusion we've got up to, we're going to assemble these NADPH oxidase enzyme complexes, and these are capable of taking dioxygen molecules, which is the normal O2 molecule, where you've got oxygen double bound to another oxygen atom. Okay, so the proper name for this sort of an oxygen molecule is to call it a dioxygen molecule, and we're going to convert it into a superoxide molecule. So let me just explain how we convert a dioxygen molecule into a superoxide molecule. So let me show you the electronic configuration, or the outer shell electronic configuration of a dioxygen molecule. So I'm going to show the electrons, the outer shell electrons of the oxygen on the left in red, and I'll show the outer shell electrons of the oxygen on the right in green. So I'll just colour this in. So in each of these covalent bonds, an electron will be coming from each one of the oxygens. So each of the oxygens will put an electron into each of those covalent bonds. So overall we've got 4 electrons in those 2 covalent bonds there. And each of the oxygens here in this dioxygen molecule will also have 2 lone pairs of electrons, as I've showed here. So there's the red ones, and here is this right oxygen atom's lone pairs of electrons. So this is the outer shell electronic configuration of a dioxygen molecule, and hopefully this is just revision from basic chemistry class. Okay, so to convert this very familiar molecule into and very inert molecule as well, a molecule that makes up 21% of the normal air that we breathe in, into a very dangerous and very reactive molecule known as superoxide, what you have to do is chuck another electron at this molecule. So let's bring in the orange electron. So here comes in another electron. So we're going to give to this molecule, and this is what the NADPH oxidase is going to do. And of course, from the name you can gather where it's going to get these where it's going to get this electron from. It's going to get it from the electron carrier reduced NADP, but we won't dwell on that, we'll just look at this. So it's going to take an electron from NADPH and it's going to give it to a dioxygen molecule. And what actually happens when you force another electron onto a dioxygen molecule? Well, what happens is one of the bonds of the double bond breaks, and you can imagine one electron from each of these, you can imagine giving the electron from each of the oxygens back to that oxygen. So imagine giving the electron from the green oxygen back to it, and give the electron from the red oxygen back to it. And then you're just going to pair this orange electron with one of those electrons that's now on its own. And this is what you're going to end up with afterwards. So here again are the 2 oxygen atoms. Now they've just got a single bond between them. Okay, so here's the electron from the red oxygen, here's the electron from the green oxygen. Now, one of the oxygens will have 3 lone pairs of electrons now, and let's say it's this oxygen on the left. So let's say it got the orange electron here, So it's now got 3 lone pairs of electrons, and it's also therefore got a negative charge. The other oxygen over here, what it will have is it will have 2 lone pairs of electrons, just as it had before, but now it will also have 1 electron that is unpaired, and that unpaired electron is not happy. When you have an unpaired electron in chemistry, that is known as a free radical, and we call the entire molecule a free radical. So this molecule is now known as a free radical, and it's extremely reactive. Free radicals are extremely reactive. I mean, even in popular culture, people know that free radicals are not good. They know that it causes aging and things like that. Free radicals are dangerous molecules. Okay? So, this molecule that I've now drawn out for you here, this is a superoxide molecule. So we call this superoxide, and we'd write this O2, and then we put a negative because indeed it does have a negative charge, and we'd also put a dot here to show that it has an unpaired electron, so it's a free radical. So superoxide is being produced by the NADPH oxidase enzyme, which we have put in our phagolysosomal membrane. We've assembled in our phagolysosomal membrane. Now, superoxide is a really good example of a reactive oxygen species. It is absolutely a reactive oxygen species. It's based on oxygen, of course, and it is extremely reactive. So it is a prototypical example of a reactive oxygen species, but not only that, it's also the basis for producing a lot of the other reactive oxygen species, and also for producing a major reactive nitrogen species, as we'll see in a moment. So it's a good example of a reactive oxygen species. So we're going to get lots of superoxide molecules being produced inside the phagolysosome, and of course the idea behind this is that these will just react with all sorts of molecules of the pathogen here and will destroy the pathogen, will cause the pathogen a huge number of problems, will cause its enzymes to stop functioning, will hopefully block its respiration, will kill it, is the idea. Okay, now, other reactive oxygen species that we can produce then, well, these come from superoxide. So superoxide spontaneously reacts with hydrogen atoms to produce hydrogen peroxide. So what you can imagine is you can stick on a proton onto this oxygen here, and you then form a single bond between this oxygen and that hydrogen atom. And then you can imagine sticking on a hydrogen atom, i.e. a proton and an electron, onto this oxygen, and then you'd end up with hydrogen peroxide. So from superoxide, we can make the other reactive oxygen species, which is hydrogen peroxide, which has this structure here. H2O2 is hydrogen peroxide. Water, but with an additional oxygen atom in it. So this is another reactive oxygen species, and this forms spontaneously from the superoxide molecules that we produce. So we don't need to have an additional mechanism to make this. As soon as we make superoxide, we make hydrogen peroxide. And just for a bit of background knowledge, whenever you hear the word peroxide, what it means is that the molecule has a single bond between 2 oxygens in it. Whenever you've got a single bond between 2 oxygens, that's known as a peroxide link. So this truly is hydrogen peroxide, because you've just got the basic peroxide structure and then hydrogen atoms off the side. So just going back to how this would form, you can imagine that if you stuck a proton onto this negatively charged oxygen here, it could use one of its lone pairs to form a single bond with that hydrogen, and what you'd have to do is bring in an actual hydrogen atom here, i.e. a proton and an electron, to bind with this oxygen atom, and that will form the other bond here. So hydrogen peroxide will form spontaneously from superoxide. And it's another reactive oxygen species, a very reactive molecule. Peroxide links are not particularly stable, so it again will react with all sorts of biomolecules and hopefully break the enzymes of our pathogen in the phagolysosome. Okay, so that's another example of a reactive oxygen species. So I've given you 2 examples now of reactive oxygen species. I want to give you one more example of a reactive oxygen species, and this comes from hydrogen peroxide. So what you can actually do is you can break down the hydrogen peroxide molecule, you can break the peroxide link, and imagine sending 1 electron back to each of the oxygens. that will then give each of these oxygens an unpaired electron, and that then forms what's known as the hydroxyl radical, which we can denote like so, OH, and then you can put the dot either after it, or you can put it in front of it if you like, because then you put the dot closer to the oxygen, which is of course where the actual free radical is present. I'm going to put it after it because I just think it looks better, but if you wish to put it in front of it, do so. So this is the hydroxyl radical. So you can produce hydroxyl radicals from hydrogen peroxide by breaking the peroxide link and sending one electron back to each oxygen, creating two hydroxyl radicals which each have an unpaired electron now. Okay, and those are also going to be free radicals, so extremely reactive. This does not occur spontaneously. There is another mechanism in the membrane of the phagolysosomal structure to actually catalyze this reaction. So this one occurs spontaneously, this one has a mechanism, has another enzyme present in the phagolysosomal membrane to catalyze it. And these are the three examples then of reactive oxygen species: superoxide, hydrogen peroxide and hydroxyl radicals, and they will all react with biomolecules and cause hopefully the death of the pathogen cell. Okay, just to make a comment now about reactive nitrogen species. So reactive nitrogen species, just as superoxide is the starting point for the reactive oxygen species, nitric oxide is the starting point for the reactive nitrogen species. So what happens is the activated alveolar macrophages, which have recognized the presence of Mycobacterium tuberculosis and have phagocytosed it into phagosomes, what they try to do, and again I'm repeating myself, but it's worth repeating, all of this that I'm describing to you now, phagolysosomal fusion and the production of reactive oxygen species and reactive nitrogen species to destroy the pathogen, this is the ideal situation. This is not going to occur very often, and I'll come on to that in just a moment. So we're going over the ideal situation. If the immune system wins, this is what happens. But this isn't usually what happens. Okay, so what's going to happen then is the phagocyte is going to try and produce induced nitric oxide synthase and sends these enzymes to the outside of the phagolysosome. So this is representing an induced nitric oxide synthase enzyme, an INOS enzyme. So INOS is a type of nitric oxide synthase enzyme, and it's specifically induced nitric oxide synthase because the expression of it is induced by the presence of a pathogen, by the activation of pattern recognition receptors on the surface of the alveolar macrophage. So this is induced nitric oxide synthase, INOS. Okay, so that's there in yellow. So this enzyme produces nitric oxide, and I'll just show you the structure of nitric oxide. It's a free radical, however it's not a particularly reactive free radical. For a free radical, it is remarkably unreactive. So INOS is going to produce nitric oxide, NO dot, which is then going to be responsible for the production of the reactive nitrogen species. However, you would not really call nitric oxide itself a reactive nitrogen species because it's not particularly reactive. So let me show you nitric oxide. So we'll have our nitrogen here, and it's nitrogen double bound to an oxygen like so. Now, let me draw that out again because I haven't really left myself enough space to actually draw the electron dot cross diagram. So we'll have our nitrogen atom here, our oxygen atom here, and we've got a double bond between them. So let's actually just go over the electronic configuration here. So oxygen, we know oxygen has 6 outer shell electrons, so it's going to have 2 lone pairs here, and 2 of its electrons are going to be involved in these double bonds. Nitrogen has 5 outer shell electrons, so 2 of them are going to be involved in the double bonds here, 2 of them are going to be in a lone pair here, and 1 is going to be on its own, and there is the reason that this molecule is a free radical. So the free radical is actually on the nitrogen. So again, if you like, you could put the dot in front of the nitrogen here rather than after the oxygen to show where it's actually present. Okay, so we produce nitric oxide molecules then by this enzyme induced nitric oxide synthase, and these molecules are going to go into the phagolysosome, and what are they going to do? Well, they're going to react with superoxide molecules to make something extremely reactive. So, what's actually going to happen then is the free radical on the nitrogen atom of a nitric oxide molecule is going to bind with the free radical on a superoxide molecule. So we've got an unpaired electron on this oxygen of the superoxide molecule, we've got an unpaired electron on this nitrogen atom here. They combine together to actually produce an extremely reactive molecule that is now going to be classed as a reactive nitrogen species. So let me now show this. So you'll have the nitrogen here, you'll have it still double bound to this oxygen, but now it's going to have a single bond to this oxygen here, and then you're going to have the other oxygen here, which will have a negative charge on it. And this molecule is written ONOO negative, like so. And it has the name peroxynitrite. Okay, so this is peroxynitrite. And it is an incredibly reactive molecule. So this is an example now of a reactive nitrogen species. So, peroxynitrite. So, from superoxide and nitric oxide, we will produce peroxynitrite molecules within the phagolysosome, and these will react with all sorts of biomolecules and cause the poisoning of the Mycobacterium tuberculosis cells, hopefully. Okay, so, to summarise then how phagocytosis works, you engulf the bacterium into this phagosome, you then fuse lysosomes with the phagosome membrane to create a phagolysosome that's going to release all sorts of horrible enzymes onto the pathogen, which is hopefully then going to lead to its breaking down. And you also set up means to actually produce reactive oxygen species and reactive nitrogen species with the hope that these will destroy the pathogen. So you build the NADPH oxidase enzyme complex on the surface of the phagolysosome, which will produce superoxide, which can then lead to the production of hydrogen peroxide and hydroxyl radicals, all of which are extremely reactive molecules. You also bring in this Inos enzyme, which will produce nitric oxide. Nitric oxide will come into the phagolysosome, react with superoxide to produce an example of a reactive nitrogen species, which I've given you here, which is peroxynitrate, a very powerful molecule. These reactive oxygen species and reactive nitrogen species, the idea is they're also going to lead to the breakdown of the pathogen. So this is the ideal situation. This is pathogen being destroyed by alveolar macrophages, which are phagocytosing it. Very few of the alveolar macrophages which phagocytose Mycobacterium tuberculosis bacterial cells will successfully complete this process and degrade the Mycobacterium tuberculosis. And this is the reason it turns into such a chronic infection. Mycobacterium tuberculosis has all sorts of mechanisms that disrupt the phagocytosis process from occurring inside the alveolar macrophages, and indeed inside of other phagocytes as well. Later on I'll talk about neutrophils being recruited to the site of infection, and they're another phagocyte, and I'll just put this key word. A phagocyte is a cell capable of performing phagocytosis, so neutrophils and macrophages are the major two examples of phagocytes. We've seen macrophages here. We're going to talk later about neutrophils, at least a little bit. So, Mycobacterium tuberculosis, as I was saying, it's very good at preventing phagocytes from actually completing the phagocytic process. It has a variety of mechanisms in order to stop lysosomes from fusing with the phagosome. It has a number of mechanisms to stop the cell from producing NADPH oxidase enzyme complexes on its cell membrane, or from bringing Inos enzymes anywhere close to the phagosome. So it prevents the production of reactive oxygen species and reactive nitrogen species. In short, when Mycobacterium tuberculosis is taken up into a phagosome in most phagocytes, what happens is you do not actually get the killing of the Mycobacterium tuberculosis. Instead, the Mycobacterium tuberculosis makes itself at home inside the phagosome, and indeed it actually starts to divide inside the phagosome. So let me actually just draw this at the bottom here. So let's have a big macrophage here. Whoops. So I'll put the nucleus over there, like so, to disguise the horrible mistake. And then let's have this as the phagosome, which contains the Mycobacterium tuberculosis. So what can now actually start to happen is, because the Mycobacterium tuberculosis has prevented the killing mechanisms, it can now start to divide inside the phagosome. So you can end up with a macrophage here filled up with a massive great phagosome with loads of these Mycobacterium tuberculosis cells inside of it, like so. Now this occurs very slowly because Mycobacterium tuberculosis actually has a really slow division time. So it takes a Mycobacterium tuberculosis cell about 24 hours, and I should put about, so approximately 24 hours to actually divide into two. That's a very slow division time compared to other bacterial cells. I mean, in other bacterial cells they can divide every 20 minutes. So compared to a lot of bacterial species, Mycobacterium tuberculosis divides extremely slowly. Okay, but the special feature of it is that it's capable of resisting phagocytosis in this way. It goes inside macrophages, it's engulfed absolutely fine, but then once it's been engulfed, it prevents the killing mechanisms very effectively. I mean, as I say, in some macrophages they will successfully unleash the killing mechanisms, and the Mycobacterium tuberculosis will be killed, but in a great majority of them it will fail. The Mycobacterium tuberculosis will win, and it will divide and divide and divide inside the macrophage, and what can then happen is the macrophage can actually end up bursting. All of the Mycobacterium tuberculosis cells will then be released, and they'll then be uptaken by new macrophages, and the cycle will continue again. Okay, so that's phagocytosis covered and how it's failing to cope with the Mycobacterium tuberculosis. So you know, alveolar macrophages, for a lot of pathogens that we breathe in, they are fantastic. They will eradicate them as soon as they arrive inside the alveoli and prevent any sort of infection from occurring. Unfortunately, it does not work with Mycobacterium tuberculosis. It's a too clever little organism. It gets the better of our alveolar macrophages and ends up surviving their attack. So what we're going to discuss in the next video is the inflammatory response, and we'll see if that can help us any further. And we'll see actually that the inflammatory response isn't going to be able to help us any further, because what's the inflammatory response going to do? It's just going to bring in more neutrophils and more macrophages. Well, loads of neutrophils and more macrophages. And they're just phagocytes as well, which the Mycobacterium tuberculosis will just get the better of. So we're only going to get this under control once we've actually had an adaptive immune response, which can help us, but we'll see that further down the line. So in the next video we'll move on to the inflammatory response, where the macrophages are going to call for help, and we'll see that.